Okay. Good morning, good afternoon, dear viewers, friends, and colleagues. Welcome to the channel Innovative Sustainability. Today, our guest is Gabriel Gemmiti, and he is joining us from Toronto, Canada. And he'll be talking about process, peculiarities, specifics about the process, and new development, new innovative solutions that are currently used. Before he begins, a brief introduction. Gabriel got his bachelor degree at University of Toronto in immunology. Then he followed his education at the Imperial College of London, where he got master degree in applied biosciences and biotechnology. He held several positions as research associate, and then he joined Sanofi. I think was 5 years now, formally, or longer, a
Around
little
that.
more
Yeah.
than that. Yes, where he started as senior scientist, then progressed to different positions, and currently he's associate director specializing in injectable projects. And he's part of the manufacturing science and analytical technology MSAT for short. Gabriel, welcome to the channel. It's a pleasure to have you here, and the floor is yours. Please begin.
Thank you so much. First of all, I just want to say thank you, and it's a pleasure being here, and really appreciate having the time to potentially share some information with everyone. So today I'll be talking about innovation in industrial vaccine manufacture. So first, I would like to go over kind of some background and some information on vaccine manufacturing at a high level. And then we'll get into some additional details around the framework of vaccine manufacturing and some case studies of some innovation in industry. At a very high level, there's three main steps. Again, these are very kind of broad brushstrokes here in terms of the vaccine manufacturing process. So we have cell culture and harvesting, which is usually referred to as the upstream manufacturing process. And the idea here is that we will take a cell, which is expressing our protein of interest, and we would start expanding it and growing it in a bioreactor. This is usually done at the thousands of liters of scale. And once we've grown our cell to a sufficient quantity, or the cell of interest to a sufficient quantity, we would then lyse those cells to start the protein isolation process. From there, we would then have our downstream, our drug substance process, which is a purification process. And so the idea is that we have to isolate and further purify the protein of interest for our manufacturing. So we want to remove all purities from the manufacturing system. So that'd be, other proteins from the cell, DNA, and other cellular components that we're not interested in. We need to remove all of that and really make sure that we have just the protein or proteins of interest from the manufacturing system. From there, we have our formulation and filling step, which are kind of more called the drug product steps. And this is where we take the purified protein that has been isolated from our previous manufacturing steps, and we combine it with other components like adjuvant and different buffers to kind of combine this all together to make a safe and effective vaccine for human administration. Once all these components are kind of combined together in the formulated state, it's ready for injection. So the next step is then we take this formulated material and we fill it into vials and syringes, and then that gets kind of shipped out, packaged and shipped out to the customer to then use for the administration. So that's a very high level overview of the kind of main steps of the manufacturing. Next, I want to just touch a little bit on the framework of vaccine manufacturing and good manufacturing practices, which is kind of the framework that the industrial scale manufacture is conducted in. So for commercial vaccine manufacture, it is definitely a very highly regulated environment, and we follow what's called GMP principles, good manufacturing practices. And so throughout the product life cycle, we have really a wide range of quality requirements that are embedded into our manufacturing process. So these really help us drive product quality into the product that we're making throughout the entire life cycle. and to make sure at the end of the day that we're providing a product that is safe and effective to the patient. To be able to do that, we need commercial manufacturing processes to initially be developed and validated to demonstrate that they're robust and reproducible. And so the way that we do that is we focus in our critical process parameters and our critical quality attributes. So for each process step, we identify a critical process or potentially multiple critical process parameters, which are tightly controlled during our manufacturing process. And then those critical process parameters go on to impact our overall product characteristics or our critical quality attributes. And that means when we do our commercial manufacturing, our routine manufacturing, we are always looking at these CPPs, critical process parameters, and CQAs to measure them and ensure that they fall within a predetermined range. So just here is a high-level example. So we have, for example, our process might have a mixing speed as a critical process parameter. It has an operating range defined as 150 to 200 RPM. And if our measured value from the manufacturer is 175 RPM, then everything should be good. And the subsequent impact to the CQA, so the critical quality attributes, would be that, for example, if our CQA is pH of the solution or the suspension, that we would expect our pH, our measured value for our pH to be within our specification of 6.3 to 6.7. So as an example, it might fall at 6.3. On the other hand, if we are manufacturing and there is an issue, for example, or we're outside of our optimal operating range of this 150 to 200 RPM. So for example, we're at 100 RPM. This critical process parameter of mixing speed may impact or is likely to impact our CQA, which would be pH. So for example, you might not have at this lower mixing speed, maybe your tank isn't homogeneous, and based off of that, your pH reading is below the specification. So you have the specification of 6.3 to 6.7, and you get a result of 6.2. And in that case, then you have a deviation or an investigation into why that had occurred and what the impact is to the product at that point. But it's definitely not the most ideal situation to be in. So this is kind of where we expect to manufacture, let's say, 99% of batches, and then we have outliers where there might be an excursion that needs to be looked at in the if that does occur. Unfortunately, we know we don't just magically start at having these parameters, very well defined of 150 to 200 RPM and 6.3 to 6.7. So there is some development that needs to go into that. And next, I'll just quickly touch on some of the methodology associated with developing these initial manufacturing.
So can I ask a quick question, maybe just
Sure.
a comment? I think for the viewers who are very familiar with systems, maybe not so new, but for those who not, just to clarify, so anytime this deviation happens, it's investigation. It's also, I think you will write CCR, right? Like control, like a change, or you may have CAPAs to address questions, and then it's all documented, has traceability, it's in quality systems, documents will reside, and so usually that can be a discussion with auditors, and they may request this kind of document. So this is not just simple investigation where you close it; it just has to be documented properly. It's just a comment to clarify for the viewers.
For sure. Yeah, no, that's 100% correct. So for any deviation we have, we would put a correction in place that should address why we had that deviation in the first place. So why we might be at 100 RPM. And as Brina mentioned, this is all in a quality system, which means there's traceability as to the investigation, what the changes were, and what the outcomes were after the change as well. And as mentioned, that can be taken a look at by regulatory agencies like the FDA or someone like that might ask for that type of information. Okay, so now we'll go a little bit into some information about making changes at scale for manufacturing. So to make changes to our commercial manufacturing process, we do need kind of some initial development and feasibility data to support the change. So we have somewhere to start with an idea of how we want to address a change that we may want to make. So kind of keeping on this idea of mixing speed, so we might have an initial development study, and in this development study, it may be performed at small scale, or we may use some sort of simulation or modeling, which is something we'll touch on the next couple of slides, where we identify potential suitable range for the parameter. So this, we might have a big range, so we test some small scale manufacture, and we identify a range of, for example, 100 to 300 RPM as suitable. From there, we have, from these studies, we can take those studies and then we move to our non-commercial batches being performed at scale. These are usually kind of called the engineering studies. And this is where we kind of tune our parameters based off the initial development study. So we take that mixing, that range of 100 to 300 RPM, and for example, we test at 100, 150, and 200 RPM, and we see how those different, the usage of those different parameters impacts our overall manufacturing process and the CQAs that we're interested at the end of the day. From there, we will further hone in on a specific parameter. So let's say from our development or engineering, 150 RPM is our best case. We get the critical quality attributes behaving or performing as we expect them to. We would then move into what's called a process performance qualification or process validation study. And this is where we take that one parameter, or we kind of lock in all of our different parameters for the manufacturing, and we validate that parameter. So we do the same run multiple times at the same parameters, and we expect the same outcome from the manufacturing system. So example here would be, we would take 150 RPM because that's what perform best, and you would perform all 3 runs at 100 RPM. And for all 3 runs, we should have the CQAs meeting our specification requirements. A quick note just on the number of runs here for a PPQ study and even for engineering run. So what we typically do for industries, we use a risk-based approach, which means that we do a risk assessment before these studies are initiated to determine what we think the risk level is with the change that's being made. And based off that risk level, we would determine potentially how many batches would be required to generate the data that we need to support the change. Similarly, there's also potential requirements for the different regulatory agencies we submit to. So for example, if we submit to the FDA or the EMA, the European Medicines Agency, or Health Canada, each of those regulatory agencies may have different requirements in terms of what they need for supporting data to support a change that's being made to our manufacturing process. That's also kind of one of the driving factors in the number of runs that we do. And then based on the change, there may be, there are quite a number of different types of studies that may need to be performed to support the change as well. So these ones here are kind of our main pathway, but there are a lot of other studies that are usually required. Some of the more common ones, depending on the type of change, might be a comparability study. So the idea here is that we have, we demonstrate the CQAs of the product before and after the change are comparable. And we may also do additional kind of non-routine testing on specific attributes of the product, so we can really deep dive into those attributes based off the change that's being made to show that they are comparable before and after the change. Following that, or in addition to that, there may also be subsequent process step data manufacturing. So what that means is we take, for example, drug substance, as shown in the earlier slides, material that's made at the drug substance step with the change, and then we take that material and we move to the next manufacturing step. So we would then move to the formulation and filling step, and we formulate and fill product based off the material that has this change already implemented, and we show that the product that comes out at the downstream or the next step in the process is equivalent to the other material that we manufactured before the change. And then finally, there's also stability data. So for that, we demonstrate that the product characteristics don't change over the product shelf life. So we'll hold the product out until the shelf life, if it's, for example, 1 year, we hold it out for 1 year, and then we measure the product characteristics throughout that year and show that they don't change or decrease over the product shelf life. And that happens with the material that has the change already implemented. So those are kind of the three additional, again, there's definitely a lot more than that, but at a very high level, these are some of the big ones for more substantial manufacturing changes. So I think that's a good framework for us to then move into a couple of examples of innovation and development during these, more focused on these development, engineering, and PPQ stages of the manufacturing.
Yes, just a comment maybe for the viewers, that stability study normally takes a longer time and you need to allocate samples for that. So it's involves logistics, specific protocols, planning, and then handling. So usually the team, different team support. So it's also by itself is a study in addition to what you just mentioned, kind of time involved and time consuming and also material consuming part. Yeah, sorry,
exactly.
just want to clarify.
Yeah, and all these studies again they're not just MSAT. Again, you're working with a wide variety of different stakeholders, and there's a lot of complexity built into a lot of these that maybe we're going over at a very high level here, but from an execution perspective, these are definitely all of them are quite time consuming and labor consuming as well in terms of a lot of effort put in. So
analysis, documentation is for sure.
Yeah, exactly. So I'll go into a couple of different case studies now. So the first one I want to talk about is what we call computational fluid dynamic studies. So for our products, our vaccines, such as diphtheria, component pertussis, and tetanus, these vaccines are usually have an aluminum basis to them. And so this aluminum-based vaccine is usually a suspension, which means that there's particulates in the or particles in the process that will settle out if they're not mixed. So what that means is that we need to maintain homogeneity throughout the manufacturing process, especially when there's high concentration of these adjuvant in our manufacturing process. So pretty much throughout our the end of our drug substance into our drug product manufacturing, we pretty much need to make sure that the product is homogeneous throughout this process. So to be able to do that, we need extensive mixing validation required for our manufacturing system and our storage tanks to show that if the product settles out, we can resuspend it, or during the manufacturing process that we don't have the product settling out as we're actively manufacturing product in it. So the opportunity here was that our typical validation usually looks at 3 runs for mixing speed and mixing volume we're looking at. And we had the potential opportunity to challenge this. And to do that, we were looking at leveraging some modeling. So this computational fluid dynamics modeling is something that can be applied to manufacturing vessels. And what it does is we take the initial inputs on impeller design, vessel geometry, and the challenge medium, aka what you're mixing or you're trying to resuspend. And with those initial inputs, we're able to build a model where we're able to look at potential shear effects and wall impact from mixing. And the model then with the model that's generated, you can then kind of play around and you can see minimum, maximum mixing volumes and mixing speeds react in your vessel of interest. So on the right here is just an example of what an output might look like. So on the top, you have strain. So you have red color here being really high strain and this blue color being low. And so as you can see, we don't want to particularly strain the product. We don't want to shear the particles that are part of our product. And so you see kind of throughout most of the tank, there's this kind of blue to bluish green color, which means we're not putting a super high amount of strain or potential shear effect on the particulates that are in the manufacturing system here. The one exception being this shaft here with these portions here. This is actually the impellers. This is actually what is actively mixing during the manufacturing process. So as expected, right around the impeller that's actively turning at a few hundred RPM, we definitely see a little bit of strain around there, but that is kind of to be expected for the manufacturing process. So overall, this profile would be kind of seen as a good profile in terms of product strain. On the other hand, we do have this other diagram here that shows what the velocity would look like in the tank as well. So again, red being really high velocity, blue being low. And what you can see is we kind of have this yellowish green color throughout most of the tank, which means that the product is moving at high velocity or high speed, which is a good thing for a manufacturer because it shows that the product is staying in suspension. What we don't want to see is really big chunks of really dark blue anywhere within the tank, so that we have, which would indicate potentially that the product is settling out during the manufacturing process. But again, same idea as before, right around the impeller shaft here, you can see this red section, which has really high velocity, again, kind of expected based off, this impeller moving at a few hundred RPM, then right around the tips of the blade here, that you would expect to see some really high velocity. But overall, again, this would be kind of what we would expect to be a good mixing profile to show that there's a good velocity or resuspension homogeneity. So the outcomes of this from the CFD modeling, we did apply it to our mobile storage tanks and our adjuvant formulation vessels. We did a simulation with both water and adjuvant as a worst case for our product. And from there, we were able to determine initial mixing speeds that could be applied to a manufacturing process. From there, we took these initial mixing speeds and we did perform confirmatory runs at the kind of min-max values as opposed to doing initial development. And we were able to move away from doing 3 runs for mixing speed to only doing 1 run as a confirmatory run based off the data that was generated from the modeling that was done. So what that meant was then we were able to go in and if we kind of look at this would be kind of the 4 corners here might be where we focus our validation on. So we would do kind of max speed and a lower mixing time. We would do lower speed and lower mixing time, high time and low speed, and then high time, high speed. And by doing this, we kind of create a box where we can show that the product is acceptable at these 4 corners. Then we have kind of a proven acceptable range in this box. And then within that box, we would then have our normal operating range. And so what that means is during our routine manufacture, we would select somewhere in a set point within this normal operating range. And that's where we would kind of manufacture our batches. If there was some sort of change in the mixing speed, we have some space where we would still be okay in terms of knowing the product quality is still ensured. So if you had a mixing speed and time that came into this portion here in your proven acceptable range, then your product would still be okay. And then, as mentioned earlier, if you kind of move outside of this proven acceptable range, you're into this situation where you have a deviation. Again, not common, but it does happen from time to time. But the idea here is that we kind of create a wider range or a wider space where we can work and play within with knowing that the product impact or the product quality is still guaranteed. And then, of course, one of the big savings or benefits of using CFD modeling here was that we had a lot of time saving and cost saving of reducing the amount of manufacturing that needed to be done with the equipment and at scale. So it was definitely a positive of using this type of model.
Just to clarify, you used it during validation, right? So basically, right, the critical step in a sense, but you still can approach it in more a clever way using this computational fluidic dynamics and select the conditions that what you want to focus on to be more targeted, more specific. And in terms of cost, how much you actually savings? Is that a month of work, 2 months of work? Just estimate.
Oh yeah, I mean it's definitely months and months of work because again, executing multiple runs per parameter. So if you do 1 run per parameter versus 3, there's definitely time saving and cost saving with the number of runs. There's also time saving and cost saving and reduced sampling and testing as well. So yeah, it's definitely quite significant in terms of time saving for this as well. And so the idea is you can start your CFD modeling quite early. Once, let's say, for example, from an engineering phase, you have, an engineering perspective, you have your tank design confirmed, you can already start working on CFD modeling. And by the time you are ready to go to validation, you've already kind of have your model to support you, and you can definitely save quite a significant amount of time from the perspective.
That's very impressive.
Okay, so then I'll go into one more case study, and this is about process analytical technology. So this is something that's becoming, or is a little bit more common, becoming more common in industry now. So PAT, or process analytical technology, is a way that we can measure our CPPs and CQAs during our manufacturing process. So PATs have an advantage because they allow a real-time in-process reading. So they give you a direct look into what's happening inside your manufacturing process as it's happening. And so this live monitoring, it's happening depending on the type of system you have. It can be happening as often as every 30 seconds, for example. It's giving you a lot of data that's about what's happening in the manufacturing as it occurs. And that information can also be directly fed into automated systems to allow kind of decision making to be made and to really increase our overall process control. Additionally, the use of PAT also gives an opportunity to remove additional offline samples that need to be collected and tested. So again, you're going to save a little bit of time in terms of testing costs and resources. So at the bottom here, just an example of one way that PAT might be implemented or might be part of your manufacturing process. So for example, if you have a manufacturing system here, and this is your probe, if you add in raw material into your manufacturing process, you expect, for example, if this is a conductivity probe, there to be a change in the conductivity of the overall system. The probe will take a reading, or is constantly taking readings, and after you add the raw material, it will indicate potentially if you've added the incorrect amount, that you get a low reading for your conductivity, lower than what the system is expecting. Based off of that, the low reading that's occurring, there is a reading sent to the system automation, and that may result in an alarm being put to the system or to the HMI for the user to be able to see. And based off of that, yeah.
I'm sorry to interrupt. I think it's telling me that we only have 7 minutes left. It should be enough, I think.
Hopefully, yeah. So then the automation makes decisions based off the reading, and from there, the automation may stop, or it may abort the batch or wait for a correction to be made. And that way we're able to kind of take a look at and address these issues as they're occurring based off PAT. So I'll go into two very quick examples of some usages of PAT in our engineering and development phase. So this is specific to adjuvant manufacture. And so we were able to leverage PAT to characterize the impact of different changes. So as we change manufacturing parameters, we're able to characterize how that would impact our process through the usage of this PAT. So we use two different PAT specifically for this. We use the FTIR probe. This monitors reaction kinetics of the process, and we used an inline focus beam reflectance probe, or what I like to call more the particle probe, and this is able to give you particle counts and particle size information from your manufacturing. So as process changes were made, we collected the data with these PAT, and we were able to see as you change one parameter up or down, there is potential impact to the product that the PAT would be able to demonstrate for us, and that would allow us to then take this information and further optimize the manufacturing process. So in the interest of time, I won't get too much into the details, but if we look specifically, for example, here, this is an outcome from the FTIR. You would potentially look at certain wavelengths that you're interested in. So for example, this might be one species in the overall, so this is different wavelengths. If you look here, this is one species, this bump is one species, and this is time. So you might look and see that this species over time is being consumed, and in the meantime, this other species is increasing in concentration or intensity during the manufacturing process. So for a potential application of this is that we might take, for example, a look at this and determine that we want to change certain manufacturing processes, manufacturing parameters and ensure that they are optimized to allow, for example, full consumption of this species, so that we have more of this species if it's the one that we're interested
Yes, it's very interesting because normally you would never catch it offline if you collect, and plus, it's not feasible to collect so many samples physically. So definitely, having probes, having real time, it's a huge advantage to
in.
see a reaction as it goes and learn from that, just like you had with modeling in a previous study. The same way you can learn much faster and plus more information to even machine learning at some point, I guess, So,
Yeah, exactly. So I think, yeah, we again, just thinking about time, we did have so yeah, I think the successes are definitely what Marina just mentioned. There's a lot of live feedback. You definitely have a better process understanding, and you have a reduction in offline testing and costs. There were definitely some challenges in terms of using some of these PAT. I think they really need to be well planned from the initial manufacturing perspective to be implemented from the very beginning. Again, there's also a lot of data that's generated from these probes, so we have to have a good way to analyze that data. And also implementing them into automation has some challenges. And then for commercial manufacture as well, working in a GMP space, there's definitely some harsh conditions that are associated with some of the manufacturing. So, we have steam in place cycles and some harsh process conditions. So finding PAT that are able to give you good readings still, but and survive kind of these more harsh conditions is definitely a challenge.
yes, it's something to, be evolved as we move on, right? Like become more
Exactly. Yeah.
robust. Yes, yes.
Yeah, there's definitely advances being made all the time on that end. I think that's mostly it for me. I'll go over a quick summary. When we develop or improve our commercial manufacturing, there are definitely unique, opportunities and challenges. We really work from a process development perspective to identify CPPs and CQAs and really make sure that they're tightly controlled during our routine manufacture. PAT and CFD are definitely things that can be further leveraged for development and validation. So CFD really gives us the opportunity to reduce our validation requirements, and PAT definitely increases our process knowledge and can assist in our overall process development. And we should really continue focusing on the implementation and leveraging these tools in the future to really benefit the biopharmaceutical industry.
Gabriel, thank you so much. That's really, lots of insights and good illustration with case studies. I think for viewers would be interesting, and some of them work specifically, so it would be, more savvy about it. Some are new to it, so it's a good educational, introductory, segue for them to learn. And again, for collaboration, those of you interested in collaboration, potentially more information, Gabriel's information would be on in description, like email. So. Connection for potential collaboration. That would
Please
be
feel free
nice.
to reach out if you have any questions or would like to discuss any further, for sure.
Yeah, that would be great. Okay. So, Gabriel, thank you so much again for joining today. I know we're kind of like almost like time is running out. Anything you would like to have as a final statement for viewers?
No, I just want to thank you again for having me, and it was a pleasure to kind of share some information. And please feel free if there's any questions or anyone would like to reach out. I'm very open to discussing further.
Thank you so much. Thank you. And our viewers, thank you so much for staying with us today. At this point, we stop. Thank you. Bye.